Hello, welcome to the second part of the Chapter 4 lecture on amino acids. So in this second part of the lecture, we are going to discuss amino acid reactions, and this includes ionization, titrations, which we talked a little bit about already, but we'll go into a little bit more detail now, as well as polymerization reactions, how they get formed into proteins, not peptides, and finally some interesting biological applications of amino acids and their derivatives. So, let's start with looking again at this amino acid structure, which we saw in the last lecture. So we already talked about how the structure, the structure drawn on the left over here has our amino group and our carboxylate group are both uncharged. And we also have mentioned that this is something that never actually occurs physiologically. However, it's a really common way that amino acids are drawn. For example, in your textbook, you might see this, or even in like the chart that I give you on your exam with all the amino acids, you'll see that a lot of times amino acids are frequently drawn this way. However, if we look at the structure on the right that has our positively charged amino group, as well as our negatively charged carboxyl group, this is a more accurate representation of how an amino acid might look at physiological pH in a cell in your body, for example. So anytime we have a molecule like this that has both a positive and a negative charge within the same molecule, but that overall has no charge, there's a special name for that type of ion, which you've probably heard before, and it's called a Zwitter ion, which is always fun to say and also fun to spell. So now that we've gone over that, we can talk a little bit more about titrating amino acids. So we've looked a little bit at this already when we talked about titrations of buffer, but now we're going to go more into detail about what it's like for the specific functional groups that are on an amino acid. So most or all amino acids have 2 or 3 titratable groups. All amino acids have an alpha carboxyl group, as well as an alpha amino group. And those are the first 2 titratable groups. And then the third potential titratable group is from the side chain. But only some amino acids have ionizable side chains. So some of them, for example, like the nonpolar side chains are not titratable, but some, like the acidic and basic ones and a couple of the polar ones, do have titratable side chains. So as we've discussed before, talking about pKa's, any group that we're titrating is going to have a buffer region that is centered at the pKa. So just to sort of explain a little bit more about that, as we'll see when we draw the titration curve, the flat part of the titration curve, where the pH stays relatively constant, the center of that part is the pKa, and that's the pH at which you have a mixture between the protonated and the deprotonated form of that particular group, whose pKa we're looking at. So for an amino acid, the first 2 titratable groups we often write with numbers. So pK1 usually refers to the carboxyl group, and that's usually around pH 2. pK2 usually refers to the amino group, and that usually happens around pH 9 or 10. And lastly, we have the pKr, which is going to depend on the side chain. Not all amino acids have a pKr because not all amino acids have ionizable side chains. And the third key point here is that any functional group, when we're at a pH that's below its pKa, is going to be protonated. And that same group, once the pH rises above its pKa, is going to be deprotonated. So when you think about it in the environment with a low pH, there's a lot of protons around, so we're going to be protonating a functional group that's below its pKa. And as the pH rises, you have fewer protons floating around, and so we're going to start deprotonating once we're above that pKa. So for an amino acid, that means for the amino group, it's going to alter between being a positively charged NH3 or a neutrally charged NH2. So, and for our carboxyl group, we're going to vary between when we're protonated being COOH and when it's deprotonated being COO minus. And something to point out is that for our amino group, when it's protonated, it has a positive charge. When it's deprotonated, it has a neutral charge. Our carboxylate group is a little bit different. Our carboxylic acid, when it's protonated, has a neutral charge, and when it's deprotonated, it has a negative charge. So that's something to keep in mind as we go through and look at these various aspects of titrations. And so that is to say, and then for a side chain, whether or not it's protonated or deprotonated, or whether or not it's charged when it's protonated or deprotonated, depends on the structure of the side chain. So some will be more like our amino group, and some will be more like our carboxylic acid group. And we'll again look more into that in detail when we start talking about the titratable side chains. So first, let's look at a general titration curve for an amino acid that has a non-ionizable side chain. So this means we're going to have just two pKa's, the pK for the carboxyl group and the pK for the amino group. So our curve will look something a little bit like this. And we can start to pick out interesting points along these curves where we have features that we know about. So first we have our equivalence points. Those are the really vertical parts of the curve. And here you can see on the x-axis of this graph, it's actually labeled in equivalence. So for every one equivalent of NaOH we added to this amino acid, we arrived at the equivalence point. So our first equivalence point is at 1 equivalent, our second equivalence point is at 2 equivalents. And then the flat parts of our curve, like we just mentioned, are going to be the buffer regions, or the pKa's. So now we can start labeling our graph. So this is our pK1, this is our pK2. Our pK1 is for our carboxyl group. Our pK2 is for our amino group. And I actually want to write that a little bit differently so it's less confusing. You'll see why in just a moment. And then our equivalence points are labeled in green. So we can start now thinking about what our amino acid might look like at each of these points of interest. So at our 0 equivalence point, all the way on the left, if we wanted to draw what our imaginary amino acid might look like, we have our N, we have our alpha carbon, we have our carboxyl group. Our alpha carbon is attached to a hydrogen and an R group. So here at the leftmost point on our graph, we're at the lowest pH. Everything is fully protonated. So our amino group is fully protonated. Our carboxyl group is fully protonated. Once we get to the first equivalence point, sort of in the middle of the graph, we've passed the point, our first pK, so our carboxyl group is now going to be fully deprotonated. So here, if we start drawing out our generic amino acid, our amino group is going to remain fully protonated because we're still below its pKa, but now our carboxyl group is going to be fully deprotonated at this equivalence point. At our pKa, we're going to have half of each. So we're going to have some COO minus, we're going to have some COOH. And our pKa is when we have about half of each of these species. At our last equivalence point, all the way on the right of our graph, we can again look at our imaginary amino acids. So we have our amino group, we have our alpha carbon, we have our carboxyl carbon. And here, our carboxyl group is going to remain deprotonated, but now we've passed the pK for our amino group. Now our amino group is also fully deprotonated, and it has a neutral charge. So when we're looking at our pK2 for our amino group, we have about half of our positively charged amino group and half of our neutral charged one. And so like we started with, this is an amino acid with a non-ionizable side chain. So it's going to have two equivalence points that we pointed out here, and it has the two pKs, which are halfway between the equivalence points. So that brings us to talking a little bit about ionizable side chains. So this is a list of all of our amino acids that have ionizable side chains, and just to reiterate, we have our 3 different pKs. So our first pK is for the carboxyl group. All of these are usually around 2. Our second pK is for our amino group. All of these are usually around 9 to 10. And finally, we have our side chain pK, and these vary depending on the side chain. So some of them, the ones that are for the acids, which we see over here, are quite low, around 4. These ones for our very basic ones, like lysine and arginine, are pretty high. They're above the pK for the amino group. We have our histidine, which is also considered a basic one, but its pK is actually very different. It's much more close to neutral pH. And finally, down at the bottom, we have our polar side chains. They are not charged. They're not considered they're not considered acids or bases. We can still just call them polar, but they do have ionizable side chains. So for tyrosine, it would be this hydrogen, and on cysteine, it would be the hydrogen in the sulfhydryl group. And these side chains, depending on whether we're above or below the pKa, will affect whether or not they have what charge they have when they're protonated or deprotonated. So for our basic amino acids, when they're protonated, they have a positive charge. When they're deprotonated, they have a neutral charge. And just to make sure everyone knows what I'm talking about, we're talking about these on the side chains, which are noted in red. For the basic ones, the side chains are off of nitrogen, right? And so nitrogen has a lone pair. It can act as a proton acceptor. It acts as a base. That's why we call them the basic side chains. And when it does accept a proton, it gains a positive charge. In the case of arginine, which is in the middle, we have this guanidino group that is part of the side chain, and in the case of histidine, we have this imidazole ring that's part of the side chain. And that's a little bit different when we talk about our other types of ionizable side chains. So for our acidic side chains, they are based on carboxylic acids, which we see over here. So carboxylic acids, when they're protonated, they are uncharged. When they're deprotonated, they have a negative charge because they are acidic, right? So they can act as proton donors. When they donate away their proton, they get a negative charge. And then that brings us to our polar side chains. So the polar side chains, I already pointed out to you which hydrogens are lost from those side chains. It's the hydrogens on the hydroxyl and the sulfhydryl groups, and those are a little bit more like the acidic ones. When they're protonated, they have no charge, and when they're deprotonated, they have a negative charge. So that brings us to what happens when we titrate an amino acid that has an ionizable side chain. So we should pick out one of these side chains and we can pick out one of these amino acids and then draw its titration curve. Each one is going to look different because the pKrs of each of those side chains is different. So as our example, let's choose histidine. So for histidine, the first pKa occurs at around 1.8 for the carboxylate group. The second pKa that we're going to hit is actually the imidazole ring at 6.04. So that's going to be in the middle, and then the last one is going to be for our amino group at 9.33. This is a little different from some of our other side chains. For example, our basic ones, their pKrs are going to be above the pK for the amino group. So the order of histidine, first this one, then the side chain, lastly the amino. For a basic one, the order will be first carboxyl, then amino, and then last the side chain. So let's go on and make our titration curve for histidine. So first we need to know the pKa's. So they're at 1.80, 6.04, and 9.33. So we can start by drawing out our pKa's. Those are going to be at the half equivalence points. So our first one, we can make a little dot right around here. Our second one, we can make a little dot right around maybe here. And our third one, we can make a dot maybe around here. So those are going to be our half equivalence points, or our pKa's. And then we need to fill in our equivalence points, and those are going to be halfway in between them. So at 1 equivalent, at 2 equivalents, and at 3 equivalents. From there, we can start filling in what our titration curve might look like. So we can try to be sort of steep around our equivalence point and sort of flat around our buffer region. And hopefully we get, whoops, I messed something up there. Let's try that again. So we have a flat point around our buffer region and a steep point through our equivalence points. That's better. All right. So now that we have our curve, we have a whole number of different points of interest. So at this point, we might want to label these points of interest and maybe make a table so we can talk about what's going on with all of them. So we'll start at this point down here, that's A, B, C, D, E, F, and G. And these are just all of the equivalence and half equivalence points on this titratable curve. So now we can start drawing out a table. So one part of our table is going to have all of these parts. And then we want to talk about each of our groups. So we will talk about our carboxyl, our amino, and our side chain. All right, so at point A, we are fully protonated, right? So we have our carboxyl group is going to look like COOH. Our amino group is going to look like NH3+. And our side chain, it's a lot of work to draw out the whole side chain of histidine, so as a shortcut, we'll just call it NH+. NH+. We are fully protonated. And from there, as we move along our graph, we can start seeing when and where those changes happen. So our first equivalence point at point C is where our first functional group is fully deprotonated. So that's point C, that's our carboxyl group. It's the first one to get deprotonated because that's the lowest pKa. Our carboxyl group is going to look like COO-. I actually just want to make it a different color so it's easier to see that. But at this equivalence point, our other side chains are all going to remain protonated still because we have not yet passed their pKs. So our amino group and our imidazole group are both going to remain protonated. At our next equivalence point 2, which is point E, that's when we start seeing a change in our side chain right around pH 6. So now our imidazole group is going to get deprotonated. We have fully deprotonated the imidazole group. We still have a negatively charged carboxyl group, but our amino group is still fully protonated. At our last equivalence point, at point G, that's when we finally have our amino group losing its proton, fully deprotonated. So now we have our amino group, neutral charge, carboxyl group is negatively charged, and our imidazole ring is also neutral. And at the half equivalence points, that's when we have some of each species, right? So at equivalence point B, our first half equivalence point, which is the pK1, where our carboxyl group, and that is where our carboxyl group is half protonated, half deprotonated. So we have some of each. And at point C, our first equivalence point, is where it's fully deprotonated. And it's going to continue to be fully deprotonated all the rest of the way through. The next equivalence point we reach is our next half equivalence point is our pK for our side chain, our pKr. That's for our side chain. And that's right around pH 6.04. And so at point D, that's where we have some of our side chain is protonated, NH+. Some of it is deprotonated, just regular old NH. So at this point, and that's sort of the dividing line where we have half protonated, half deprotonated. So we can also sort of fill in the rest of these. So we know that it's still protonated here, and then it's going to be fully deprotonated past that equivalence point. So last, let's talk about our amino group. That's our last pK. It's our pK2, and it's for our amino group. So at point F, our half equivalence point for our amino group, we are going to have half of our protonated species, NH3, and half of our deprotonated, NH2. And again, we can fill in the rest of this, and the whole amino group is protonated the whole time up until we reach this point. So what does this tell us? It can help us figure out what the charge is on our whole amino acid at any of these given points. So in a sense, we can sort of add up and make a total for our table that we just created. So at point A, we have 1 neutral and 2 positives. We have a charge of +2 for this amino acid. At point at point C, we have 1 negative and 2 positives, so we're at +1. At point E, our next equivalence point, we have 1 negative, 1 positive, and 1 neutral, so we are at 0. And at point G, our last equivalence point, we have 1 negative and the rest neutral, so we're at -1. So that's great for our equivalence points. What about our half equivalence points? We have sort of some of each, right? It's a little bit messy. So here is where we can sort of have a half, right? So we're somewhere between, for point B, we're somewhere between +2 and +1. So we are probably somewhere around 1.5. And that doesn't mean that any one molecule has a charge of +1.5. That's not possible. But we have some molecules that are +2 and some molecules that are +1. So on average, the molecules average out to be +1.5. Same idea for point D. We're between 0 and +1. So we have an average of +1/2. About half the molecules are +1 at this point, and about half of them have no charge. And lastly, at point F, we just are going down to the negative, so we are at -1/2 for the same reason. So what's interesting about this is that now we have identified a point at which our amino acid has a neutral charge. And we call this the isoelectric point, or the pi. And we know that in this case, it's point E, which is our second equivalence point, which we know is somewhere halfway between our pKr, right, and our pK2. So to figure out our isoelectric point, we can average together these two pKs. So if we average together our first one, 6.04, plus our second one, 9.33, and we divide by 2, we can arrive at our average. Which is about 7.69, if I did that correctly. So you all can check my math. And yeah, so that is our PI, the isoelectric point. It's the average of the two surrounding pKs from that point. So that brings us to, so there's a lot going on in this molecule, right? But once we can sort of break it down, we can figure out where the pKs are, where the equivalence points are, and what that means for each of the functional groups. And once you break it down into those pieces, you can figure out some interesting properties about the amino acid. So let's talk about what happens when we want to join together amino acids. So we all know that amino acids make up proteins, and to do that, we had to join them together in polymerization reactions. So this type of reaction is essentially a condensation reaction or a dehydration, where the two individual amino acids are joined together from end to end. A water is removed, and you form what's called a peptide bond, which is just a sort of specific word for an amide bond. We've seen amide functional groups before, so let's look a little bit more closely at that. So our first amino acid over here, it has our R1 side chain. The carboxylate group on there is going to combine together with, so the carboxylate group of one amino acid is going to join with the amino group of our second amino acid. The atoms marked in red are going to be lost as water through this reaction, as we see here, and then we're going to be joined together with this peptide bond. And we've talked about what an amide functional group looks like. This is an amide bond. We call it a peptide bond in the context of a protein because it's these bonds that help turn amino acids into peptides. And when short chains of amino acids are often just called peptides, and longer ones are called polypeptides, and a very, very long one is often what is usually what we call a protein. And peptides are always named or sequenced in the same order. They always start from the N-terminal end, the amino end, and they always end with the C-terminal end, or the carboxyl terminal end. So unlike for DNA and RNA, we often will write a direction of 5' to 3' or 3' to 5'. It's the convention for proteins to simply write their sequence, and you can always assume we're starting with the N and we're ending with the C. So in this case, if we wanted to name this dipeptide, right, we have two amino acids put together, we have a dipeptide, and the sequence would be whatever R1 is, followed by whatever R2 is. Because the backbone of all peptides are the same, with the N, the alpha carbon, and the carboxyl carbon, what differs is the side chains, and that's how we name them. structure of a peptide. So first, we want to trace the backbone. So the backbone of our peptide comes from the individual amino acids that make it up. So we start with our amino group that's bonded to the alpha carbon, and that's bonded to the carboxyl carbon. And so that's our first one. Our second amino acid starts with our amino group bonded to our alpha carbon here, followed by the carboxyl carbon. Our third one starts here with our amino group bonded to our alpha carbon, bonded to our carboxyl carbon. And our last one starts with our amino group, goes to our alpha carbon, ends with the carboxyl carbon. So how do we identify the other important features here? We have our N-terminal end, that's over here, and we have our C-terminal end on the other side. So we know that this peptide with 4 amino acids in it will always be read from N to C. So in this case, it's written from left to right. And in between our amino groups and our carboxyl carbons, we have our alpha carbons. Our alpha carbons, as we show here, we have this first one, which is attached to our first side chain. We have our second alpha carbon over here, attached to our second side chain. Our third alpha carbon is attached to our third side chain. And our fourth alpha carbon is where our fourth side chain comes from. And before we move on from there, I also want to point out where the peptide bonds are. So the peptide bonds are in between, basically, where the individual amino acids were, as you can sort of deduce from what we talked about before about how the individual amino acids are bonded together from the C terminus, the carboxyl carbon of one to the amino group of the other. So this is our first peptide bond, this is our second peptide bond, this is our third peptide bond. And understanding where the backbone is and where the peptide bonds are can help you figure out how many amino acids we have, as well as counting how many side chains there are. So we can see here we have 4 side chains. We traced out 4 individual amino acid backbones, and we connected them together with 3 peptide bonds. So the number of amino acids we have here is 4. And now that we've identified where they're located in this peptide, we can now figure out the sequence of this peptide. I won't ask you to memorize the structures of the side chains. You'll be given the side chain structures in a table on exams or quizzes. But I do expect you to be able to recognize and match them when you see them. And it's important to be really careful when you're matching them because a lot of them look really, really similar to one another. So let's start with our first one on the left. So if we look at some of our table of amino acids, this one is isoleucine. So it has the structure where you have these 4 carbons that are attached together with the second carbon, which is chiral, attached to the alpha carbon of the amino acid. So that's our first one. That's isoleucine, which you can also call ILE or just I. Our second amino acid, which I have circled in yellow over here, we can match it up with methionine. Methionine is nice to identify. It has this sulfur in the middle of this hydrocarbon chain. So we know that our second amino acid is methionine, which is also called met for short, or just M. Our third amino acid looks like it has an ionizable side chain, right? It's negatively charged. So let's take a look at these. So we have 2 acidic amino acids on the right-hand side of the slide here, and they look very similar to each other, right? They have a carboxylic acid on the end, and then they have either 1 or 2 carbons that attach them to the main body of the amino acid. So in this case, we have glutamic acid because we have 2 CH2s followed by the carbonyl, the carboxylate, so that's a total of 3 carbons. And that's what we see over here, 1, 2, 3 carbons. So that's glutamic acid, which you can also call glutamate, especially when it's charged like this. And the abbreviation for that is GLU or E. So be careful with those abbreviations, right? Because not all of them are just the first letter of the amino acid. And our 4th amino acid side chain that we can recognize is a polar one. It has a hydroxyl group. And so that one is going to be, oops, I forgot to paste in that slide, but this one is serine. And serine has a CH2 next to a hydroxyl group. And so serine, its 3-letter code is SCR, and its 1-letter code is S. So, and you want to be careful not to mix this one up with something like cysteine, which looks very similar, except instead of in a hydroxyl group, it has a sulfhydryl group. So that is the sequence of our amino acid. You could write it as IMES. You could write ILE, MET, GLU, SEER. You could write out the full names. You could write out isoleucine, methionine, glutamic acid, serine. Those are all correct. It's just important that you correctly identify the side chains, like I said, because it's easy to mix them up with one another. So let's talk a little bit about some amino acids and amino acid derivatives that have interesting biological functions. So first we have dopamine over here. So dopamine, you might notice, looks very similar to tyrosine, and in fact it's derived from tyrosine through a few different reactions. So, and it functions as a neurotransmitter. So it is biologically active in our brains. It transmits signals from one neuron to the next. The next interesting biologically active amino acid we have over here is histamine. Histamine looks very similar to histidine. And so you can see it has that same sort of imidazole ring. We have our sort of backbone, but you can see instead of being attached to what would be our alpha carbon, this imidazole ring is actually attached to the 3rd carbon, which doesn't have a carboxyl group. So that part of the structure is very similar to dopamine up here. There's no carboxyl group anymore, and the side chain has sort of shifted over by 1. So the function of histamine is it's really important to trigger inflammation. It's often something you hear in the context of allergy and allergic reactions. And the last biologically active amino acid we're going to talk about is glutathione, which are shown here on the bottom, and which is often abbreviated GSH. And it's derived from a tripeptide of glutamate, cysteine, and glycine. And its function is to reduce reactive oxygen species. So let's talk a little bit more about that. So the way it reduces reactive oxygen species, it comes from the similarity to cysteine in the middle, where cysteine has that sulfhydryl group, which is just like the center of this glutathione molecule. If you have 2 glutathione molecules, just like with cysteine amino acids, you can have an oxidation reaction where you form a disulfide bond in between the 2 glutathione molecules. So you would have GSH and HSG, and then you'd combine them together to get GSSG. Very similar with this disulfide bond. And something interesting to note here is if we try to trace the amino acid backbone of this structure, here's our 3rd amino acid, that's glycine. So we have our amino group, our alpha carbon, and our carboxylate group. The middle amino acid, like we just talked about, is cysteine, with our amino group, our alpha carbon with the sulfhydryl and CH2 and our carboxyl group. And then first we have our glutamate group, glutamate amino acid, but this one's attached a little bit differently. So with the first way I just mentioned, the peptide bond here is between the carboxylate carbon from the backbone and the amino group. With these two, the peptide is actually being formed from the side chain part of the glutamine. So over here we have our amino acid backbone with our amino group, our alpha carbon, and our carboxylate part of the backbone. The side chain of glutamate also has a carboxylic acid, and that side chain is actually what's participating in this peptide bond over here. So that's something interesting structure-wise about glutathione. And the last sort of biologically interesting function of amino acids I want to mention is in green fluorescent protein. So green fluorescent protein was originally found in jellyfish. It's an aspect of bioluminescence. It helps them glow in certain conditions. And the way that it has fluorescence to it is the functional part of it, as we see on the top, is made with 3 amino acids. You have a serine, a tyrosine, and a glycine. And the sort of central part of that we want to look at is the tyrosine. So tyrosine starts out with this nice aromatic ring, and when it undergoes a cyclization reaction, then we form a more extensive ring structure that has even more extensive conjugation of all of these double bonds, and so that allows for a color change essentially, and so then we end up with this nice green fluorescent color. And this is something that not only do we see in nature, we also can bring it to use in the lab. So you can see on the bottom, green fluorescent protein has been inserted into one of the proteins in the skin, and now some of these mice have skin that is fluorescent green. Green fluorescent protein is a really helpful tool in the lab. It's a relatively small protein, and you can, using lab techniques, you can attach it onto other proteins or other biologically relevant things that you're interested in looking in, and make whatever protein you want to look at fluorescent. So you can use this, for example, for microscopy to make a protein you're interested in turn green, and you can see where its specific location might be in a cell or in a tissue. And you can also use it, these aspects of fluorescence, for a number of different lab techniques that we won't go into right now. But with that being said, now we've talked about a number of different functions of amino acids in terms of ionization and titration, as well as the condensation reactants and peptide bonds, and a little bit about biologically relevant molecules. So you're ready to go ahead and answer those practice questions.